think so.
Let's see, now what happened to my,
Oh, did yours stop?
To maintain the organization of the chromosome. And in the Mero lab, one of our main goals is to understand how bacteria coordinate the initiation of replication and segregation. And to do this, we use Caulobacter crescentus. Caulobacter crescentus is a great model for cell cycle studies, but we use it as a model for my project for a couple of reasons. The first one is that Caulobacter initiates replication only once per cell cycle, and that allows us to track when one cell has initiated replication and what the cell cycle after that looks like. Another great technique that we have in Caulobacter is that they're easily synchronizable, and so that means that we can start our experiments using a homogeneous population of cells that is going through the cell cycle all at the same time. So now how cells maintain the organization of the chromosome as cells replicate and segregate their chromosome still remains unknown. But I told you that chromosome replication initiation and segregation are concurrent in bacteria. This is how they happen in Caulobacter. In the chromosome, we have the origin of replication, and 8 kilobases away, we have a centromere region called PAR-S. We have a protein PAR-B, which is part of the PAR-ABS system, that binds to PAR-S directly, and I'll be referring to this as the centromere from now on. Replication then initiates at the origin of replication through DNA-A, the canonical replication initiator. But it is not until PAR-S is replicated that segregation actually occurs. And segregation is done by PAR-A, which is the active chromosome segregator that pulls the centromere to the opposite pole. And that's done by this gradient. So at the end, you have a cell with 2 centromeres anchored at each pole. And our work so far shows that these 2 events, replication initiation and segregation, are highly coordinated, and that coordination is done through DNA-A and PAR-A. So now when we zoom in a little bit on that region in the chromosome between ORI and PAR-S, in bacteria that encode both of these regions, they're always found in close proximity. Increasing the distance between these 2 regions in Caulobacter results in 6 cells. And as I told you, segregation does not occur until PAR-S is replicated, even when put further away in the chromosome. So one thing that we notice is that when we disrupt the levels of PAR-A, there is an increase in replication initiation in Caulobacter, which I've told you it's not typical because they replicate only once per cell cycle. We're still trying to figure out how PAR-A does this, but it seems to be through DNA-A. Interestingly, another thing that we notice is that DNA-A also binds at the PAR-S locus. And when DNA-A cannot bind to these sites, there's also dysregulation of replication initiation. We're still characterizing how these 2 regulators drive these events. And we'd like to use goldfish to visualize what these 2 regions look like as replication and early separation of the chromosome occurs. So that's one of the questions that we have. Another question that we have goes just to the anchoring of the centromere to the cell pole. So the chromosome is anchored at the cell pole by interactions between the protein that you will see labeled here in cyan, which will be PAR-B indicating PAR-S. And it's anchored at the pole by a protein that I have here in yellow, which is called POP-C. And these 2 proteins anchor the centromere to the pole. So now how the centromere is actually anchored and then released is not really known. Because if we look at this little movie, we have one centromere or one ORI gets replicated and then gets moved. But there's a little bit that it got stuck, of course, where there is a little bit of separation before there's replication. And so we don't really know whether PAR-S gets replicated and then separated from the anchoring pole, or if it gets duplicated first and then separated. We can't answer this question yet with our current resolution. And it will be great if we could use goldfish to learn what is the order of replication and separation from the cell pole. We have some clues that it could be.
Stephanie, can maybe, well, you know, our speaker has a plane to catch, and has to leave at 3:15.
Okay.
So,
well, here's the questions that we have. So the first one is, how do the genes find the address inside of the cell, and what is the trajectory of these genes? What is the organization of ORI and PAR-S at the cell pole? And also, we can visualize the anchoring of PAR-S to the cell pole and the release to initiate replication of the chromosome. Thank you so much.
Thank you very much. And this is obviously an invitation for Powhatan to return to help with this, push this project forward. So we're going to hear now about genome maintenance by one of the developers of the technology of goldfish, not to be confused with the aqueous version of it. Okay, thank you.
Thank you very much. Thanks to QCB for inviting me and giving me this opportunity to represent our lab. Today, I want to talk about two stories of CRISPR-Cas9 technology that we developed in the lab, and we use them to, and how we use them to study DNA repair, and how we use them to develop a genome editing tool at high resolution. So ever since the CRISPR-Cas9 has been demonstrated the potential to perform programmable genome editing tool, this field has been growing pretty fast. And soon enough, it led to a Nobel Prize a few years ago. So I'm going to briefly talk about how we actually use it to make it as a technology platform. First of all, we need to somehow introduce the Cas9 and RNA complex into the cell by either extrapolation or genetic encoded expression. And this complex, once enter the cell, they would search through the genome, searching for the palm site. Once they find the palm site, it binds to DNA. If the sequences that next to the palm site matches the RNA sequences within the complex, then the Cas9 will perform the cleavage. After Cas9 leaves the site, it leaves a double-strand break. This double-strand break will then trigger endogenous DNA repair mechanism to this cut site. So by hijacking this mechanism, people have been using this tool to develop genome editing platform or applications such as genetic screening and other applications. Specifically, we want to use this tool to study DNA repair and perform genome imaging. So the first story that I want to talk about is very fast CRISPR. This is actually the project that was published before even I joined the lab. So here I'm just trying to present the technology developed in the lab. But so far, we still have a lot of new ongoing projects that use this kind of technology. So I think this will be a good topic to talk about. So if we want to use Cas9 system, CRISPR-Cas9 system to study DNA repair, briefly, what we need to do is we want to introduce the Cas9 into the cell. After the Cas9 complex perform the double-strand break, as I briefly already mentioned, it triggers the endogenous DNA repair mechanism at the break site. There are potentially two outcomes. One is that if the repair was successful, then the sequence will become exactly the same sequence as before, and the repair is done. Or sometimes some error could be introduced. For example, random deletion or insertion will be introduced to the break site, and this cell the sequence will become different from its original sequence. In case of successful repair, the freely floating Cas9 complex will still find the same targets and cut. So if the repair was successful, this cell will actually undergo many rounds of cutting and repair cycles. So as you can imagine, if you want to use this technology to study DNA repair, there's a challenge. The challenge being that we have a population of cells, and we incubate those cells, say 20 with the Cas9 complex for example, 24 hours. What happens is that we got a heterogeneous population. So we don't really know when the cutting happened in a cell. We don't really know when the repair happened. So it's hard to really monitor repair events using this method. So previous postdoc, Yang, and previous MD graduate student, Roger, came up with the Bing Wu's lab in Johns Hopkins. They had this idea. So what if we can control the Cas9 cleavage activity at high spatial temporal resolution? So what we can do is then synchronize the Cas9 cutting in a population of cells. Once the cutting is synchronized, then the DNA repair mechanism is also synchronized. So now we need to take a step closer into how Cas9 function. So as I already briefly mentioned that the Cas9 and guide RNA complex, after introducing into the cell, they look for the POM sequence, which is usually NGG sequence, 3 nucleotide. So once it finds the POM sequence, Cas9 will actually open up the DNA proximal to the POM site. And previous work and also some work from HALAB verified that only 9 base pair was required for Cas9 complex to form stable binding on the DNA. So after the stable binding was formed, the Cas9 will continue to unwind, to open the DNA double strand, and only 16 base pair matching from DNA sequence to the guide RNA sequence. It triggers the conformation change of Cas9, and it performs a double strand break. After the Cas9 left the site, there will be a double strand break. So, as we know that the conformation change of Cas9 of a protein happens at a very fast time scale, the sub-second time scale. So really, this entire process, the rate limiting step, is the Cas9 searching step. So the idea Yang had was that, what if we introduce a photocleavable K-gene group at the second half of a guide RNA, which will actually block the Cas9 cutting, but it won't affect the Cas9 binding. So what we can do is that we can incubate this complex with the cell for sufficient amount of time. So for example, in 12 hours. So after this sufficient amount of time, most of the target site will be bound by this complex. And then if we introduce UV light to remove the block, it triggers a conformation change. It immediately move on to the next step. Then the cell will be cut at the same time. So this is exactly the idea of very fast CRISPR. As an analogy, essentially, we can think of like Cas9 is a scissor, and we give it sufficient amount of time for the scissor to find the places we want to cut in the genome. After the scissor finds the cut site, it won't cut because there's a block. After we trigger the UV light to remove the block, as soon as we remove the block, then the scissor will cut the genome. So some of the data shows that, this very fast CRISPR can actually generate double strand break very efficiently compared to the previous methods. And it happens even within seconds. So this is just a quick comparison. The very fast CRISPR can generate double strand break in a very high specificity, and the activation time is actually pretty fast compared to the other way of generating double strand break. So this makes it a very good tool to study DNA repair for double strand break.
Excuse me?
Mm-hmm.
I missed, maybe you mentioned that you're talking here about all organisms, or this is only aimed at like just culture cell or something? I missed the context. Which organisms is this built for and working in?
We perform this in human cells, mammalian cells. I think it works in mouse cells as well.
We have talking culture cell, or you're talking about anything in the organism?
Culture cells.
Just culture cell.
Yeah, by far. Yeah, so now with this very fast CRISPR tool, what Yang did was that we want to see if we can use this tool to monitor the DNA repair in real time in living cells. And again, this is U2OS cells. So what Yang had was he introduced 2 fusion proteins into U2OS cells. One is the Cas9 GFP. The other one is a repair protein 53BP1 fused with anchory. So the idea is that this Cas9, he designed a guide RNA that targets a specific site. So this Cas9 would actually sit on a specific site. But because this is a live cell, so there's a lot of freely 14 Cas9 GFP in the background. So it's hard to really see a single GFP signal. So what he did is that he actually introduced another truncated RNA, which only has 11 nucleotide in a guide RNA. So it actually can form stable binding, but it won't perform a cleavage. And he used this truncated RNA to target the repetitive site that is 36 kb away from our actual cut site. So we can use this site as a reference site. It actually forms a bright puncta on the fluorescent microscope. So this is one of the sample movies. So you can see there are green puncta. This is the signal from the reference site referencing our actual cut site. And as soon as we trigger the UV light, the double strand break will be, Cas9 will perform a cleavage activity, and we can see the repair protein will enrich at the cut site in the red channel.
Question.
Yeah.
So what power of UV light are you generally having to use, and you induce other damages to the cell DNA?
It's actually pretty gentle. Just by the fresh light, UV fresh light could activate the entire plate of cells. Yeah.
Sorry, say again, which cell type is this?
U2OS.
And
It's human
that is,
cells.
So remind me, which type of cell is that? What's the origin of that cell?
It's a cancer cell.
So does it make
I
sense?
think the bone cancer, the bone cell.
So why does it make sense to look at DNA repair in such an abnormal cell? Is DNA repair not abnormal in a cell like that?
I think different cell model have a slightly different DNA repair behavior, but why they choose this to begin with, I don't know. But it seems to me, actually in our lab right now, people use this technology to study mouse embryonic cell, also in hack cells, other type of human cells. I think it works pretty well. Equally well.
I see. Okay.
Yeah.
Okay.
Yeah.
Thank you.
Right. So yeah, you can see from the single cell tracking, we can track the cut site, the signal, the reference site, and also the repair protein. And you can see pretty clearly that as soon as after the cutting, the activation of very fast CRISPR, within 3 minutes, we can already see the enrichment of the repair protein at the cut site. So, which means that the repair signal is actually very fast. It's within minutes. And we also observe that there are many rounds of cutting. For example, you can see there's a first round of repair, and the repair signal goes away, and then there's a second round of repair occur, which is like what I briefly introduced before, because the 3D14 Cas9 is still within the cell. So after repair, if repair was successful, that piece of sequence can still be targeted by other Cas9. So there will be a subsequent round of cutting and repair. The interesting observation here is that the first round of repair usually takes a longer time compared to the subsequent rounds. We don't know why. So this is one observation. The other observation is that we found that the two alleles within the same cell seems to have a correlated repair durations. You can see the repair durations from one allele is somehow correlated to the second allele. Right. So far I have shown that this very fast CRISPR has this very high sequence specificity because we use the Cas9 CRISPR-Cas9 system. We also can activate the very fast CRISPR within a very short amount of time. The next thing we want to test where if we can actually activate this very fast CRISPR locally within the cell. Essentially, we want to see if we can actually activate this DNA repair or cleavage activity at a single locus. So one experiment that Yang did was he designed a guide RNA that target 200 repeats of a repetitive sequence. And we want to use focus UV beam at one of the allele, but not the other. So the expectation is to see the repair protein will accumulate around one of the allele, but not but not the allele that we don't activate. So this is one of the example movie that if we only shine UV light on this allele, we can see there's an enrichment of the repair protein, but not the other one. So to further test the capability of this method with the single Cas9, so he used exactly the same system where he designed a guide RNA target only one specific sequence, but he downstream this specific sequence, we have a cassette of the Cas9. We can use it as a reference point. And this is another example showing that only one of the allele is actually activated and triggers a repair mechanism. So
Can I ask a quick question? I know you're representing a TAKIP and he wanted to have you talk about this.
Right.
But please leave enough time to talk about, is this understanding this fast CRISPR, is that going to be essential for understanding how the goldfish works?
I think these are two separate techniques, but they both use a CRISPR system. Yeah,
Okay.
I will talk about the goldfish work in a few slides. Yeah.
Okay, so I think that is the, since, I mean, this is beautiful work, don't get me wrong. And he's fairly well known since that science paper on this fast CRISPR. But as I said, I really would like to have as much time as possible about the goldfish.
Okay, sure. I think we are done here. So yeah, I think he wants to show that because we have a lot of follow-up projects with this and also with this technique combined with goldfish. So yeah, so here we show that this technique has the capability to activate the single the cutting at a single allele as well. You can see this, it has this high specificity. It has this right. It can activate like 50% of the cutting with the repetitive sequence, and roughly 10% successful rate at a single Cas9 cutting. So this is the quick summary. This very fast CRISPR provides second level control of Cas9 cleavage, and we can use it to monitor the repair mechanism, repair process in a living cell. And it also has the capacity to perform a single allele activation. Yeah, and as I mentioned, there are many ongoing projects in the lab to improve these methods, also using this method to study deeply into the DNA repair. So the second topic that I want to talk about is goldfish, which is also the project I mainly work on. So this project is really motivated by a question where we have this 2 meter long linear genome packed into a tiny 10 micrometer in diameter nucleus, and how does this genome function? And apparently, genome is actually organized in a very hierarchical manner. Specifically, we would like to study the problem that happens at a few hundred nanometers range and from few kb to MB level length scale. Why do we interested in this scale? Because apparently DNA repair, it has been shown to happen at this range. And other interesting biological processes such as enhancer promoter interactions, which lead to transcription, happens at this scale as well. And we are also interested in using the technique that we developed to study the VDJ recombination. And it also happens at this scale. So the most popular method to study genome architecture is by using this conformation capture technique. Essentially, we fix the cell and then we ligase the DNA. We digest the DNA and then we ligase. So, and then we pour the piece of DNA down and send it for sequencing. So we'll be able to figure out which pieces of DNA will actually incur close proximity when they were in fixed cells. And this method has been quite effectively map out the organization of the chromosome from the larger scale domain to smaller topological associated domain, and to finer scale where there's a interaction of the loops. Combined with some modeling and simulation work, people have been suspecting that loop intrusion is actually an active mechanism to maintain the finer scale structure. Alternatively, people were also able to use imaging method to study chromatin structure. For example, we can use a sequential rounds of DNA fish method to map out a structure. What we can do is that we can image one locus at a time, and then we wash out the probe and introduce a second round of image, and then wash out the probe. So step by step, we will be able to map out the structure after the reconstruction. The good thing about the imaging method is that it has this multi-model information. For example, we can label DNA and RNA at the same time. One of the very beautiful work done by Alistair lab. I'm not going to go into detail about this, but they were able to correlate the transcription activity with the chromatin structure in a tissue-specific manner. So at this point, we were able to really correlate the chromatin structure and the transcriptional output. But there's actually a missing piece in this picture, which is the protein. Because the protein, such as the SMC protein, can actually regulate the chromatin structure. For example, the cohesion or CCF really contribute to the final scale of chromatin structure. And also, transcription factor can regulate the transcription as well. So it would be good to include the protein into this imaging scheme as well. However, the field has been moving forward to include the protein imaging with this DNA imaging. But it has been quite limited because most of the most recent work, they have demonstrated that the protein imaging is actually really difficult. They're only limited to these larger structure proteins. Mainly because if we want to do traditional DNA fish, we need to use heat to denature the DNA for the fish probe to hybridize. And this heating process actually is pretty harsh for protein imaging. So I'm going to quickly summarize the limitation of traditional DNA fish method. It requires heat treatment, which not only prevents quantitative protein imaging, but also people have been showing that heating the cell can perturb the fine structure of chromosome. And the traditional DNA fish method, because heating is not sequence specific, it actually opens up the entire genome. So what happens is that it increases a lot of non-specific binding and increase the background. And also, if we heat up the DNA to open the two strands, the complementary strand is still there. So there's a chance for the complementary strand to hybridize back to the pocket strand, which could compete the DNA fish probe hybridization. So it lowers down the label efficiency. Both of the high background and low label efficiency prevents us to perform high resolution genome imaging.
Sorry, I lost the one point. But I thought that's what Alistair did, didn't he? In Alistair's case,
yeah,
he did do the latter. I mean, high sort of genome, sort of visual mapping or whatever you want to call it, right?
Right. He mapped it, but he was not able to see protein at the same time.
Oh yeah. I thought your last point was not about protein. I thought the last point was about that you can do high resolution imaging. I thought that was the last point. Maybe I misunderstood.
Right. So yeah, so their resolution is down to 2.5 kb, 2 kb. They cannot do finer. Our goal is going to see if we can do sub kb level resolution.
Got it.
Yeah. Thank you for pointing out. So yeah, the field actually have recognized this issue, so that people have been trying to figure out ways to overcome the heat treatment or the limitation of traditional DNA fish. This is, for example, catfish utilizing the Cas9, which only binds to DNA, but it doesn't really cut. And this is a work from Robert Sinner's lab where they fuse this Cas9 with halotag and fluorescent dye. It has been demonstrated that this method is pretty effective in terms of like labeling repetitive sequence, such as telomere, and also repetitive sequence in a single gene. However, it's pretty hard to use this method to see non-repetitive sequence because the lack of the signal amplification. So previous graduate student, Yan Bo, he actually has this brilliant idea. So at the time, the lab is actually interested in study variants of Cas9 protein. And there's a specific variant of Cas9 protein. It's a mutant. And what it does is that instead of perform double strand break, it only cuts a single strand. And this single strand will actually be exposed. And at the same time, also in the lab, actually in collaboration with Yang, they have figured out that a helicase rep in a close conformation, which we call rep-X, has very strong processivity, and it targets the 3' overhang. So what we can do is that after the Cas9 incubation, we can treat the sample with rep-X. And rep-X will actually target this 3' overhang and unwind the DNA, which generates the single strand region for us to hybridize with the DNA fish probe. So this method has been quite successful. You can see compared to the Cas fish, the signal to background ratio is like 10-fold improvement. So
Sorry,
yeah.
Back to that plot again. So
Right.
how many this is in oligo or this is a Nick translation, your probes?
oligo.
So how many oligos are you designing for it?
This one, specific one here is 57.
And you're comparing with a catfish that has a single dye of the same type, is that right?
Right. This catfish, I think it has something around 70 Cas9. And the
Oh, this is a repetitive sequence.
non-repetitive
So
sequence.
why is it not a single Cas protein with the dye there?
Non-repetitive. So they have to use 70.
Oh, it's only have multiple guide RNAs.
Yeah. Multiple RNAs.
So-
70.
So if you put 70, why does it not give you
Because the
not 70 is too much to ask, but why does it not give something comparable to your 60 probe?
because the background is also much higher. We can go back to the
In the catfish?
yes.
Why is that?
Why
You
is
have the
that?
specificity, sorry, of the guide RNA, right?
Right. So DCAS9 is actually known that the specificity for DCAS9 is there's much more off-target events for DCAS9 compared to
To the real one,
the
the wild type one, or whatever.
the ones that actually cut.
Oh, I see.
Cleavage has a higher specificity.
Okay, I didn't know that. Good.
Yeah. So the background is actually higher in the catfish.
So it's not really the fault of the lack of amplification, because here he's trying to do amplification in your catfish, and you're still not really getting it.
So here we only need to use 9 Cas9 to target the similar.
Right. But I'm saying in the catfish, the problem is not really lack of amplification.
Right.
That doesn't help because you're doing 70, you say, so you're trying to amplify 70 per one side, right?
Right.
And you're still not.
Right. But I'm saying like the amplification from the oligo pen is actually higher.
Yeah.
You can pack in denser probe in a shorter region. Yeah. So yeah, this method actually solved two of the questions in the traditional DNA fish because rep-X locally unwind the DNA, and iterate is actually local. Cas9 achieves the local denature, so we don't need to suffer from the high non-specific binding of the background. However, this current goldfish method is still not perfect, and it introduced extra problem. So when when rep-X unwind double-strand DNA, the other strand is still there. So the question of like what the other strand is doing, right, is still unclear. It could be hybridized back to the original strand, so they compete with the DNA fish probe, which lowers down the label efficiency. Also, at the time of the development, for this Cas9 to work, there's a specific fixation has to be used. So this fixation method requires 50% methanol and acetic acid. So this acetic acid is actually denaturing the protein. So the goldfish is still not compatible with protein imaging. So even though it doesn't use heat treatment, it still doesn't compatible with.
Why do you need it? As best you understand? Why do you need nasty fixation condition?
Yeah, this is exactly a point that I'm going to address. Yeah. So yeah, having in mind that I want to develop a high resolution DNA imaging methods. So I took on, this is where I pick up the project. And actually, we actually figured out another way of achieving local denature. But instead of using RAPX, we are using exonucleus, and we term it goldfish X. So I begin with the observation that there are a few DNA fusion methods that actually utilize the exonucleus to generate a single-strand DNA. And I found that these methods, they were actually compatible with high fixation, which is using this chemical cross-linking. And so I thought of like, maybe I can just use this exonucleus to generate single-strand DNA instead of RAPX. And can I just simply replace the RAPX with exonucleus and hoping the protocol to work? It would have been good that if it worked out that easy. So I simply swap out the RAPX in the protocol with exonucleus, and it actually didn't work. We actually didn't see any DNA fusion spots. So we actually sat back to think about what's going on. So as I mentioned, that we have to treat the cell to achieve local denature. We need to treat the cell with a Cas9 variant, which cuts only single strand, and it actually exposes this 3' overhang. And there are actually evidences showing that after the cleavage, Cas9 might still be there. So there's a possibility that Cas9 is actually blocking the access of exonucleus to this 3' overhang. So at the same time, there was actually our lab has a collaboration working on a protein which is called helicase transcription factor, and this is short for HLTF. So this protein targets 3' overhang, and it has a little bit this translocation activity, just enough to kick out the Cas9. So we thought of like, if we use this HLTF treatment after the Cas9 incubation, we might be able to start to see DNA fusion signal. So here I'm going to show a few examples, which is actually the case. So after HLTF treatment, and we started to see some DNA fusion spots showing up in our signal, in our sample. However, it kind of worked, but the label efficiency is really, really low. So we step back again to think about what's actually going on. We actually realized that XO3 and exonucleus that we use, it actually digests the nick, but it didn't really digest the 3' overhang. So there's another class of exonucleus digests the 3' overhang to the front end. So we thought of like, if before the exonucleus treatment, we insert a step where we use a kernel fragment to digest the 3' overhang, then we might be able to get better signal. And it actually worked out pretty well. After the sequential treatment of these 3 different proteins, we finally see good DNA fusion spots. As a control, that if we don't use HLTF, we don't really see good DNA fusion signal.
Wait, can you explain what we're seeing here? So the purple is your dye, or the purple is just your hux or something?
Sorry. So this is just the dye. So the purple, this is just single. I have the nuclear channel, but I didn't show. So the purple is the dye. This is one of the first set of experiments, so the signal to background is really bad. I have much better signal after.
So the signal I'm seeing, remind us again, you have, that means these are oligos that remain throughout your nucleus?
These are the
background purple.
These are the background, yeah, the oligos. Yeah.
So they don't get washed, so that means they bind to stuff, is that right? Is that how you?
So this batch of oligo was actually not as good. There are some free dyes that flows in the background. And yeah, I think later time we actually figured this out, and we were able to clean out the background pretty well. Yeah.
So I should just be impressed that this has more spots in it,
Right.
bright spots
Right.
than on
There
the
are more-
previous slides.
Exactly.
So then that's why you say the efficiency is much greater.
Yeah, that's true.
Okay,
In
but now
one,
you're gonna go on and help us get rid of, or
all
improve the
that.
background.
Yeah, you'll see that. Yeah. Okay, so here I just want to show that the new method actually solve one of the problem that was left by the previous goldfish, which is that actual nucleus actually generates real single-strand DNA templates. But still, we want to see if we can really replace these nasty, fixation methods by more commonly used paraformaldehyde fixation, which is compatible with immunofluorescence. So I actually tried to run the goldfish protocol with the PFA fixed cells. It didn't work. We didn't see any DNA fish spots in those cells. These are the U2OS cells again. But when I started to titrate or play around with different level of fixation, sometimes I started to see the DNA fish spots. So after a few rounds of optimization, we actually realized that the permeabilization of the cell nuclear membrane is pretty important because Cas9 has a huge volume. So if we don't really permeabilize well the nuclear membrane, Cas9 don't really go into the nucleus. So this is actually the data that you should be impressed.
Okay.
We have been, going through cleaning up the probes and everything. If actually, I can probably try to.
Now, this is
Make
pretty
it
good.
bigger.
This is clear. Seeing is believing.
Right.
So, but what's your ground truth here? How many copies of the locus do you have?
This is a very, good point. So for beta-actin, it's actually pretty crazy. This is U2OX. We actually do some statistic. I think the mean is like 5, 6.
So this is again one of the cancer cells.
It's one of the cancer cells.
So again, tell me what's the logic, right? I mean, you need your ground truth, right, to interpret your
Yeah.
fish.
We have
so
some-
why wouldn't you do a case where you know the ploidy exactly?
Okay, there are two.
Crazy cancer
There are
cell.
two reasons.
Okay.
The U2 is crazy cancer cells, easy to grow. It's super easy to grow and easy just to do some quick experiments. And also it's flat, so it's also easier to image the nucleus.
But am I right that you don't have a ground truth about the copy number?
Yeah, so for this specific locus, we don't. But there's another locus that we do, which is the next slide. So yeah, in this case, we use it to stain another gene, which is much well behaved, right? 4 spots, as you should expect, or 2, as you can see. We do some, just some analysis. Yeah. You can see the background signal is very well separate from the background, and this is really as good as it gets. 2.3 kb, 83 probes. This is the standard of the state of our method compared to the state of our methods. So non-repetitive sequence, so we can use it to label any sequence, essentially any sequence that we want.
Now, you guys are single molecule people, so how many probes do you have found? How many fluorophores?
Yeah, we're trying to figure this out. I think the label efficiency here could be around 50%, but we are still in the process of figuring this out. And we want to improve this label efficiency. Yeah, for sure. So now that we have optimized the protocol for it to work with the PFA fixed cells, the real question is still remains, right? Can it work with the protein imaging? And the answer is actually yes. So with this, series of optimization, we were actually able to combine the DNA fish method with the immunofluorescence imaging. This is just to demonstrate that this 2 protocol works. There's no correlation between the DNA locus that we label and the protein that we label. There shouldn't be any colocalization or anything. So just to show that for larger structure proteins such as nucleolus, this is the POL1, so you should see that enriched in the nucleolus. It works pretty well. For the DNA repair protein that we are super interested, it also works pretty well. You can see the very bright puncta of this protein, and at the same time, the DNA fish spots. So the next thing that, as I mentioned in the beginning, we really want to push the resolution with this method, right? So before I was showing that this is 2.3 kb, roughly close to the state of our imaging resolution. People use like 2 kb, 2.5 kb, so it's roughly there. We want to see if we can see with much fewer dyes or much shorter segment that we label. Again, this is U2OS cells. We use 26 probes. We can see clear DNA fish spots. with a little bit drop in the label efficiency. Not all of the cell has 4 spots. Want to push further to see how far we can really push this technology. We shrink it down to 300 base pairs. We can still see clear fish spots with a little bit compromised label efficiency. So we're still in the process trying to optimize this protocol. Our goal is trying to see if we can really label the DNA segment around 500 base pair with this method.
So can you explain why is that sort of an important goal to go to, let's say, sub-KB solution?
Sub-KB resolution. So if our goal is set at roughly 500.
Why is that? What makes that number sort of something you're after? That's what I'm trying to understand.
Even why? Okay, so
What is the feature you're after that you need to go to 500 and like the 2 KB or 5 KB is not, doesn't get you?
yeah. So our goal is trying to see if we can really capture chromatin looping. And chromatin looping, the higher the resolution, the better, right? The better you can visualize the loop. Because when looping happens, actually DNA could come together. So 2 KB might not be sufficient for you to map out the shape of the loop.
And formaldehyde doesn't mess it up? Formaldehyde does not mess it up?
Oh, this we don't know.
But I thought the high C would know that, right? With the high C, they've done, I think, both with and without fixation, right? So they would know if the fixation messes it up or something, right?
High C always, I think you always have to fix it with paraformaldehyde. So high C is like the way that we can compare our data to. It's a gold standard, right?
And they remind me to what resolution nowadays?
For high C is like KB level. People do micro C, which can push maybe a bit around KB or maybe even shorter.
So what will be your ground truth when you beat the high C then? That's
If we are doing higher than high C, then I think we have to have different control. I think the ground truth, because one thing is that imagine we do get to see single cells, and high C don't have the single cell data. So we need to control.
I thought there is high C single cell, no?
But the signal is really, really bad. You don't get to see KB level, probably not even 10
Okay.
KB level. Yeah. Okay. Yeah. So with that, in any case, I think we need a separate control for the single cell data. Yeah. Right.
But I thought at the end of the day, one of the things you're interested in, at least you said at the beginning of your talk, is being able to get the DNA structure. And I'm not just talking about the loops. You have various chromosomes there. I don't know what chromosome that particular gene is sitting on.
Right.
But how far do you think you're from that goal of being able to say, well,
See the structure.
chromosome 1 is over here, this part of the nucleus, chromosome 2 is over here, chromosome 3 is over here.
If you just want to identify chromosome, I think that's actually a much easier task because we just need to label the sequences specific to each chromosome, right? But if you want to see the entire structure of chromosome, I think that's a harder task, right? And I think everybody has this a good point because if you want to see the entire chromosome, then maybe we don't need such a high resolution, right? Lower resolution could also already give you some sense of where chromosome 1 is and where chromosome 2 is, right? So because we actually want to use it to study DNA repair. So that's why our immediate next step. Now we already kind of settle this method. We want to extend this method to do sequential imaging so that we can map out the fine structure of a chromatin. And then we want to couple this with a very fast CRISPR so that at the end, we can actually generate a double-strand break and fix the cell after, for example, 30 minutes, 1 hour, 2 hours, 3 hours of the very fast CRISPR activation. And then we want to map out the upstream and downstream chromatin structure. At the same time, we want to image the repair protein so that we want to see the distribution of repair protein along the chromatin structure. We can use this tool to study DNA repair.
Wait, so the Cas doesn't mess up the chromatin structure?
The Cas?
The Cas9, the protein, right? In all it's doing, I always in my head imagine that it messes up the chromatin structure because it has to, open things and all that.
At which step? at the very fast CRISPR step, or
No, in your,
goldfish.
yeah, I mean, like, I'm with Anne, I'm in the goldfish
Yeah.
mind. So you talk about looking at the finer structure of the chromosome using the goldfish, right?
Yeah, yeah,
So when you do goldfish, when you do the cast opening,
right.
are you not messing up the chromatin structure?
So this we actually don't know. But when we open up the chromatin, it's already paraformaldehyde fixed. So I would imagine you could. So we have to compare this to maybe high C, right?
Even just the fact that you fix them and then you can do all this biology on them is always surprising and alarming to me. The same with RNA fish, right? That you fix them and then you can do all these things that involve removing things from one another. It's just odd, I think.
Right. Yeah. Yeah, and as one of the purpose of this trip for me to come to QCB, we actually really hoping to extend our new goldfish method, goldfish X, to other organisms as well, including bacteria and yeast. This is just like some of the. He's actually sent me some of this preliminary data was done by a previous graduate student. Yeah, it has the potential actually to do it in yeast as well. So. Yeah, with this, I want to thank you for your attention. Special thanks to my mentor, TJ, and brilliant graduate student, Tian, that I've been working with. And really, I think I have to thank QCB for the funding and support for this work. So with this, I think I will take questions.
Yeah, I mean, I know we asked already a lot of questions, but I think that, you know, maybe go for the easier goal because people talk about trying to use all, you know, ChIP-seq, Hi-seq, just trying to get an idea of the 3D arrangement of the chromosomes. And they even look like for chromosome interactions, and then chromosome interaction with the nuclear membrane. So if you could get very quickly to snapshots of where the chromosomes are, you know, in any instance of the cell, because it seems to me like with some of them, if you do it on the Calibacter, of course, that's, you know, they can synchronize those cells. That would be interesting.
Right.
You know, I think, but I'm not for sure, the yeast, you can also synchronize them. So if you just did that and then showed snapshots of where the DNA, where the chromosomes are, that would already be a big step.
Right.
Yeah, I know everyone talks about DNA repair, but you know, let's figure out first where the chromosomes are sitting.
Right. Sure.
They
Yeah.
just draw cartoons and nobody really fits them into these to the nucleus, at least of the eukaryotic cell. And you said you thought that would be an easier thing to do.
Yeah, just mark out the position of chromosomes.
Yeah. And if you do it at this resolution, you know, and then you can do it single cell, you're really ahead of the game of everybody.
Yeah, sure.
Are there other questions from the audience?
One more, I got one more. First, again, rarely I want to again agree with that because I think, sure, you guys may have strong motivation for DNA repair because, you know, maybe cancer money's there or something, whatever, but
Right.
there's so many other things in the unperturbed cell that are worth just knowing better. So on that, and especially if it's QCB money, then might as well, you know, that's not for cancer stuff. But maybe you just mentioned, so does the method play nice now with RNA fish, which is nice and easy by itself, but is usually doesn't play nice with DNA fish?
We haven't tried.
You should. It's,
Yeah.
you know, it's an easy and robust method, and it also gives you good sort of ground truth for, you know, if you look at the transcribed gene, the RNA fish exactly tells you where it is, right?
Yeah.
And so you get a, you know,
Yeah.
and then you put in a minflux and you save the DNA and the RNA and you have yourself your, you know, what do you call it? Miller Christmas tree.
Yeah.
No, but it is it's true, his comment. I mean, I know many groups do work on cancer cells. And they're not even real, in my opinion, real cancer cells because they've been immortalized. And they don't even look at live cells, primary cells, because it's hard to culture them. Those are all problems that are being addressed in the SDC. I've asked various groups to work on that, and I think they're finding a way to get the primary cells, because then we can do 3D imaging of them quite quickly. But that's the next step to figure out what's going on in any of these organelles. And, you know, through the work of Lieberman Aiden down at Rice, he sequenced every animal in the Houston Zoo and has been published. So you pick your favorite animal, you know, and you can get crowdsourcing for it if you want, right? But I'm serious, there's so many, but it is more difficult to handle like either primary cells in a human. I do realize this, and I still haven't heard a consistent algorithm or protocol for you know, because they have a turnover that nobody likes, so they'll even immortalize primary cells. So it's something that we want to do, and as soon as the group is able to achieve that, then that's some information we can also share with ACIPs.
Right, sure. Yeah.
But you know, Ito, the Cancer Institute is also interested in doing the comparison to primary cells, because they immortalized everything, and they don't know what that chemical modification really does to the cell. Right. Yeah. And at least on a 3D level that they can visualize easily. Right. So I think this is a fantastic technique, and I like it because it's single cell. And I think there's so many possibilities, but don't make the task too tough for yourself.
Right.
To pick something easy, right?
Okay.
Right.
Yeah.
If there are no more questions, maybe we.
I have one question. Just
Okay.
one quick-
Wait, when's your flight? No, we better, now.
Yes, leave at 3:30. The car will pick him up downstairs.
Okay.
Yeah, please.
Okay. So I just wanted to ask about RepX. I know that you've moved on from using RepX, but do you know how many kilobases does it unwind? And also going off a little bit of what Ito said, how many kilobases can you really unwind before you lose the confidence that that's what the structure looks like?
Right. So for the first question, RepX, I think in vitro experiment tells us if roughly 1 kb, 2 kb at a kb level. Then the question would be like, when we swap it out with the exonucleus, what's the persistivity of exonucleus, right? We have tested like using just single Cas9 and targets, and we follow up by say 2 kb of probes. The signal, actually the efficiency for single Cas9 target is already kind of low, roughly like 20%, 30%. But once it targets, actually we can see signal, I think matching the signal what we got from roughly at the kb level. So I think it roughly at the order of like few hundred base pair to kb. So what we decide is that to make sure we don't have this issue, say within the 2.3 kb region, we actually use 9 Cas9. So spacing each of them roughly 300 to 400 base pair apart. So just to make sure that this coverage is sufficient for us to create enough space for the fish probe to target. Yeah.
You have a question quickly?
Sorry for this question, but you immobilize DNA and you do something on it, like make modifications and mobilize it again, put it wherever. In this process, do you ever go in a loop of, okay, you fix something, but you created some repair, like a problem somewhere, like you might have damaged DNA in the process of doing something, and do you measure that, or is it like, okay, we did our stuff and we just put it back, we don't care what?
Yeah, potentially we could damage the DNA. We could, but I feel like the damage that we introduce with all these proteins is probably gentler than just heat the cell up to 90 degrees and try to break everything apart, right? So I feel like our method is still gentler compared to the current state of our DNA fish method.
But have you tried ever measuring that?
As I said, yeah, measure we have to benchmark with others. We haven't really got to the point where we see the structure yet. So that's definitely something that we have to do.
Okay, so if there are no further comments, let's thank him, so he'll come back again.